Imagine a patient, right? They've just been diagnosed with bladder cancer.
A really tough diagnosis.
Yeah, and the oncology team decides to go in heavy. They use a neoadjuvant approach, which means chemo before surgery.
Right. Usually a combination like gemcitabine and cisplatin.
Exactly. So they get the gemcitabine and cisplatin, and in some of these patients, the response is just phenomenal. I mean, the tumor mass practically melts away.
Which is always the goal, yeah.
But then in other patients receiving that exact same regimen, the exact same drugs, the tumor barely budges. It just sits there.
And historically, we've always assumed that's because of the cancer cells themselves.
Right. The immediate assumption is always, oh, there must be some intrinsic resistance mechanism, like a genetic mutation that builds a shield against those platinum-based agents.
But what if that conventional wisdom is, well, what if it's looking in the wrong place entirely?
Yeah, what if the difference between a total cure and total failure has like less to do with the cancer cells and literally everything to do with a covert population of bystander cells, cells that the chemotherapy just sort of accidentally wakes up?
It forces a complete paradigm shift, honestly, in how we approach tumor biology. Because for decades, the focus has overwhelmingly been on the malignant cells.
Which makes sense, they're the cancer.
Right, but by doing that, we're effectively ignoring the complex ecological neighborhood that those cells actually live in.
Which creates this massive diagnostic and therapeutic blind spot. So that is what today's Deep Dive is all about. We are zeroing in on a recent preview article in Cancer Cell by Mahadevan and Kalluri.
Yeah, and they're dissecting this incredibly fascinating, highly complex study by Mar and colleagues. It really upends our traditional understanding of how chemotherapy actually functions in the body.
And our mission today is to completely unpack this research. We are going sentence by sentence, looking at every single cellular mechanism, every genetic model, and every clinical trial they mention.
Because the implications here extend way beyond just bladder cancer. We are looking at a fundamental reevaluation of how systemic therapies alter the entire tumor microenvironment. We are not skipping any details until this is crystal clear for you.
Okay, let's unpack this. To really grasp the magnitude of this discovery, we have to look closely at the battlefield itself, right? The tumor microenvironment, or TME.
Yeah, and the dominant stromal cells in that environment are fibroblasts.
Now, in a normal, healthy physiological state, fibroblasts are basically just your standard response to tissue injury. They heal wounds, they deal with tissue fibrosis.
Exactly. But in oncology, we're dealing with cancer-associated fibroblasts, or CAFs.
Right. And CAFs are tricky.
The crucial thing to remember about CAFs is that they aren't just like passive structural elements. They actively regulate localized immune cells. They constantly remodel the extracellular matrix. They dictate blood vessel growth, even nerves.
Let's use an analogy to help picture this. Think of fibroblasts like the body's construction and repair crew, right? You get a cut, they show up to build the scaffolding and fix the wall.
That's a great way to look at it.
But in cancer, it's like a hijacked construction site. The crew gets corrupted, and a large portion of this crew is forced by the tumor to build defensive walls. They suppress the immune system. They literally physically block the chemo drugs.
We call those tumor-promoting CAFs, or TP-CAFs.
But scattered in that same site, you have this rogue faction of the crew. They're actively rebelling, trying to tear the tumor's defenses down from the inside.
And those are the tumor-restraining CAFs. TR-CAFs. This dichotomy is just essential to understand.
Because if we look at the specific single-cell RNA sequencing data in bladder cancer, you see all these different subsets, right?
Yeah, it's a very heterogeneous population. You have AIR-CAFs, which are interferon-regulated. You have MSAFs, which are myofibroblast-like. You have RESCAFs, the resting ones.
And then crucially, you have ICCAFs, the inflammatory CAFs.
Right. And the data on those ICCAFs paints a very grim picture. High infiltration of inflammatory CAFs consistently correlates with a poor prognosis.
Bad news all around. Aggressive disease progression, higher rates of recurrence.
Yeah, because ICFs are prolific secretors of cytokines that actively suppress T-cell function and promote malignant growth. They are the quintessential tumor-promoting phenotype. The bad guys.
But wait, hang on. I need to push back on this a little.
Sure.
If fibroblasts are just the scar tissue of a wound and the tumor is driving them toward this inflammatory, bad ICF state, why would introducing a systemic poison like cisplatin help? Wouldn't a toxic drug just trigger more tissue damage, more inflammation, and therefore more of these harmful ICFs?
And that right there is the exact physiological paradox that makes this study by Ma and colleagues so brilliant. You would totally expect a blunt, cytotoxic insult to just trigger widespread inflammation.
Right, because it's a poison.
But a tumor isn't just a static collection of mutating cells. It functions almost like a rogue organ. The stroma is dynamic, so the stress induced by the chemotherapy doesn't just kill cells.
It does something else.
Yeah, in specific subpopulations, the chemo forces a radical metabolic reprogramming. It literally flips their phenotype.
Wow. Okay, let's just pause here for a quick 5-minute recap to consolidate what we've established for you so far.
Sounds good.
So, fibroblasts heal wounds, but in tumors, they become CTAFs, hijacked fibroblasts. CHAFs are a mixed bag. You have tumor-promoting ones and tumor-restraining ones, and the inflammatory ICCAFs mean very bad news in bladder cancer.
Perfect summary.
So here is a question for you, the listener, to ponder. Think about a time you got a deep cut that scarred. Now imagine those exact same healing cells going rogue to protect a tumor. How would you even design a drug to stop them without stopping your body's innate ability to heal?
It's the ultimate tightrope walk in oncology.
It really is. So if CCAPs are this mixed bag, how does chemotherapy alter the balance? Let's move from the background to the specific clinical evidence that Ma and colleagues actually discovered.
Right, so to figure this out, they utilized an incredibly rigorous clinical methodology. They didn't just look at random cross-sectional data across different patients.
No, they used single-cell RNA sequencing on paired human bladder cancer biopsies.
Yes, and that word paired is doing a lot of heavy lifting there.
It means they took a biopsy from a patient immediately before neoadjuvant chemotherapy and then sequenced another biopsy from that exact same patient right after the treatment.
Exactly. It allows for true longitudinal tracking of how the cells evolve within the same host.
Yeah.
And the transcriptional shift they observed was just striking.
So what did they see after the chemo?
Well, the single-cell resolution revealed the emergence of a distinct, previously unrecognized subcluster of CCAFs.
And these newly emerged cells had a very specific surface marker, right?
Yes, PTGER3. They were highly expressing PTGER3.
Okay, so PTGER3 positive, but what does that actually mean they were doing?
Well, when the team performed gene set enrichment analysis to figure out their function, the transcriptional signature was unmistakable. These fibroblasts had undergone a massive metabolic shift toward lipid utilization.
Lipids, so they were basically obsessed with fat.
Heavily enriched for fatty acid oxidation, omega lipid oxidation, and long-chain fatty acid metabolism. They were just burning through lipids at an incredible rate.
And because of this really unique metabolic diet, the authors coined a new term for them. They called them lipid oxidation CAFs, or lipoCAFs.
lipoCAFs. And what's fascinating here is where the clinical translation becomes undeniable.
Oh, the patient cohort data.
Yeah. The researchers took this specific lipoCAF transcriptional signature and that PTGER3 expression, and they cross-validated it across multiple independent human patient cohorts.
And what did it show?
The presence of these PTGER3 positive lipoCAFs strongly and consistently correlated with a favorable prognosis.
Which is just crazy. It forces us to look at neoadjuvant therapy through an entirely new lens. I mean, we designed chemotherapy to be a toxic bomb aimed at rapidly dividing malignant cells.
A total sledgehammer.
Right. But this data shows it's actually acting as a highly precise biological switch. The chemo is actively remodeling the tumor's neighborhood to spawn a population of good tumor-restraining cells that help the patient.
It fundamentally alters the therapeutic objective. The efficacy of the platinum-based regimen isn't just about direct cancer cell death anymore. A huge portion of its success actually relies on its ability to recruit the patient's own stromal tissue into the fight.
Okay, let's hit our 10-minute mark with a quick mini recap.
Let's do it.
So Ma and colleagues looked at before and after chemo biopsies. They found a new cell called the PTGER3 positive lipoCAF. These cells love metabolizing lipids and fats, and having them means the patient has a much better prognosis. So here is the question for you to think about this time. If a toxic chemotherapy drug can accidentally create a highly beneficial tumor-restraining cell, what does that tell you about how blunt our current cancer tools are?
It tells us we have a lot to learn about pulling these biological levers intentionally.
Exactly. And to do that, we have to understand the molecular mechanism. We know these lipoCAFs are the good guys, but how do they actually fight the tumor?
Right, we need to trace the exact biological algorithm step by step, from C8F all the way to the immune system.
This is basically translating figure 1 of their paper into a vivid story. Let's start with the mouse models.
So the researchers co-injected PTGER3 positive CAFs alongside bladder cancer cell lines into mice. And what happened was it suppressed tumor growth and created a T-cell dominant tumor microenvironment.
But the CF itself isn't directly attacking the cancer cells, is it?
No, no, it's more like a specialized manufacturing hub. Deep inside the lipoCF, the first critical step is an enzyme called CYP1B1.
CYP1B1. And they proved how necessary this enzyme is using a really cool genetic model.
Yeah, a fibroblast-specific deletion. They crossed CYP1B1 FF mice with PDG FERC mice.
Right, so using that Kralock system with the PDG for promoter, they deleted CYP1B1 only in the fibroblasts, leaving the rest of the mouse totally normal.
And when they did that, the stromal defense collapsed. The tumors grew way faster, and the chemotherapy response failed completely.
Okay, so CYP1B1 is clearly the engine. What is it doing inside the lipoCF?
It's oxidizing a substrate called arachidonic acid, or AA.
And when CYP1B1 oxidizes arachidonic acid, what does it make?
It creates a specific oxalipin called 11-HET.
11-HET. Can you give us the full name just for the record?
Sure, it's 11-hydroxyacetyltrinohysic acid.
Beautiful. Okay, so 11-HET is created. What happens next?
Well, 11-HEP is secreted out of the CF. It acts as an endogenous ligand, basically floating through the extracellular space until it finds its target. An effector, CD8 positive T cell, the immune system's assassin.
Here's where it gets really interesting. Let's use an analogy. The CIF isn't a soldier fighting the tumor. It's an armorer, and it's manufacturing a biological lockpick. That lockpick is 11-HET.
I love that.
So 11-HET sneaks over to the immune cell, the T cell, and uses that lockpick to disable a critical safety switch.
And that safety switch is a molecule called PTEN.
So how does the lockpick actually disable PTN?
Physically, 11-HET displaces PTEN. It causes it to aggregate and lose its localization on the cell membrane.
And why does PTN's location on the membrane matter so much?
Because PTN is a negative regulator. It's the brakes on the immune cell. So when 11-HET displaces it, you're essentially cutting the brakes.
Hitting the gas.
Exactly. Inhibiting PKN activates PIP3 signaling at the membrane.
PIP3, and that triggers the downstream cascades, right?
Yes, the massive buildup of PIP3 triggers the AKT and MTORC pathways.
AKT and MTRC. And what is the final output of that cascade?
The T cell basically goes into overdrive. It pumps out massive amounts of effector cytokines, specifically interferon gamma or IFN gamma,
Yay.
and tumor necrosis factor alpha or TNF alpha. And at that point, the T cell becomes hyperlethal to the cancer.
It's a fully automatic weapon against the tumor. That algorithm is just incredible. The chemo stresses the CIF upregulates CYP1B1, which turns arachidonic acid into 11-HET.
Yep.
11-HET enters the T cell, disables the PTEN brakes, activates PIP3, AKC, and MTORC, and boom. The T cell unleashes IFN gamma and TNF alpha.
It's a masterclass in intercellular communication.
Okay, 15-minute mark. Let's recap this pipeline.
Let's do it.
CYP1B1 turns arachidonic acid into 11-HET. 11-HET enters CD8 positive T cells and disables PTN, which is the brakes. PIP3, AKT, and MTORC pathways activate. And T cells unleash IFN gamma and TNF alpha to kill the tumor.
Exactly right.
So, listener, knowing this precise chain reaction, here's a question for you. If you were a drug designer, at which exact step in this pipeline would you try to intervene to boost the immune response?
That is the million-dollar question in pharma right now.
Seriously, which brings us to a really glaring issue. If we know CCAFs are this powerful and we have this exact mechanism, why aren't there CCAF targeting drugs in the clinic right now?
Well, to understand that, we have to walk through the clinical graveyard of failed trials.
The clinical graveyard, yikes.
Yeah, it's historically been a disaster because we didn't understand the heterogeneity. We didn't know about the good CCAFs versus the bad CCAFs.
The researchers were just targeting all fibroblasts indiscriminately?
Exactly. Take pancreatic cancer, for example. It has a super dense fibrotic stroma. So early trials try to dismantle that barrier using sonic hedgehog or SHH inhibitors, drugs like vismodegib and sonitigib.
Vismodegib and sonitigib. The theory was break down the wall, let the chemo in.
Right. And there were some transient improvements in drug delivery at first, but ultimately it led to vastly enhanced tumor aggressiveness. The trials were totally terminated.
Oh, wow. And what about fibroblast activation protein, or FAP?
Same story. They tried a drug called AMD 3100 targeting FAP-positive fibroblasts in pancreatic cancer, and it just caused massive systemic toxicity.
Didn't they try going after the extracellular matrix, too?
They did. They used ECM-modifying agents like PEG-PH20 to target hyaluronidase. It completely failed to show any clinical benefit.
What about TGF-beta signaling?
They used losartan for that. It showed really promising preclinical results in mice, but the clinical benefits in humans were totally unclear.
And vitamin D analogs?
Lactobacillus, again, failed to show significant therapeutic benefits.
So if we connect this to the bigger picture, the biological misstep here is obvious.
It is. All these trials failed because they used a sledgehammer instead of a scalpel.
Right. They wiped out the bad TPC-CAFs, the inflammatory IC-CAFs that we talked about earlier, which is good. But at the exact same time, they completely wiped out the good TRC-CAF.
Including our newly discovered lipoCAFs. They destroyed the host's innate defense system.
Which brings us to our final 18-minute mini recap.
Go for it.
Past trials targeting sonic hedgehog, FAP, the ECM, TGF-beta, and vitamin D all failed, or literally made the cancer worse. And they failed because they killed the tumor-restraining good guys along with the bad guys.
Exactly.
So listener, here's your question. How do we selectively enrich PTGR3 positive lipoCAFs without accidentally feeding the inflammatory IC-CAFs that cause cancer recurrence?
That is the defining challenge for the next decade of research.
So what does this all mean? Zooming out, the paradigm shift here is massive. The tumor microenvironment isn't just a static background or just scar tissue. It's a highly dynamic ecosystem.
It is. And treatments like neoadjuvant chemotherapy don't just poison cancer cells. They can physically force host cells into a highly specialized, lipid-burning, cancer-fighting state.
It's incredible. But before we wrap up, I want to leave you with a final, unaddressed question from the text to ponder, something to really mull over.
Well, this is a good one.
If we know the exact molecular lockpick is the lipid 11-HETER-D, could the future of cancer treatment bypass the fibroblasts entirely?
I mean, theoretically, yes.
Right. Could we simply synthesize 11-HETER-D in a lab and deliver it as an adjuvant drug alongside chemotherapy?
Yep.
We could instantly supercharge the T cells to attack the tumor without even needing the CFs to manufacture it for us.
It's a completely brilliant therapeutic vector to explore.
Well, thank you so much for joining us on this deep dive. We really hope you walk away looking at chemotherapy not just as a poison to cancer, but as an active architect of the immune environment.
And keep questioning those medical dogmas you encounter.
Absolutely. Because the difference between a tumor-resisting treatment and melting away might just be a secret population of bystanders waiting for the right signal to fight. See you next time.